Welcome to Monobiology. In this little mini lesson, we're going to look at the questions you folks had to answer in your designing a controlled lab experiment. And we're going to specifically look at questions 13 through 18. So you're going to want to get those handy and get those ready. What we're going to do quickly is we're going to do question 13, and then hopefully you can quickly go through the rest of them. We're going to quickly go through the rest of them. I'm going to stop at the ones that are more difficult, and I'm going to kind of give you some strategies that you can use, hopefully, when it comes time to do a quiz or a test for this stuff. Okay, so the first one we're going to read really carefully. A group of students hypothesizes that the amount of alcohol produced in fermentation depends on the amount of glucose supplied to the yeast. They want to use 5%, 10%, 15, 20, 25, and 30% glucose solutions. The questions asked are A, what is the independent variable? B, what is the dependent variable? C, what control treatment should be used? And D, what three variables should be standardized? Now, you can see the answers are in there, and that's fine. But what I'm going to show you is how I would solve this particular problem. Okay, notice up here, in this area here, I've indicated that the independent variable, the IV, is going to be a purple color, and down here I've indicated that the dependent variable is going to be an orange color. I'll use those colors throughout this lesson to help you kind of figure this out. So remember, the purple is independent variable and the orange is the dependent variable. So I'm going to start with the independent variable. I'm going to change my color to purple here. Okay, so a group of students hypothesizes that the amount of alcohol produced. All right, so this word right here, produced, that's a key word. All right, produced implies that that particular thing is happening towards the end of the experiment. If that's the case, then the amount of alcohol, this part right here, has to be the dependent variable. But look what I just did. I circled it in purple. I'm going to go back here. Look, I made a mistake already. I'm going to go back here. I'm going to undo that. I'm going to change my color back to orange. And just remember what I said. The word produced, that's a key word. And the amount of alcohol, therefore, has to be the dependent variable. Okay, so you have to, when you read these things, you have to kind of look for key words that stand out, and that's one of them, produced. That tells you that that particular thing comes towards the end, which means that's what you're measuring, which means it's the dependent variable. Okay, so if that's the dependent variable, it says depends on the amount of glucose. Well, the amount of alcohol produced depends on the amount of glucose supplied. Right? And then it even goes one step further. It says they want to use a 5, 10, 15, 20 glucose solution. So right here you can even see that the experimenters in this case are changing the amount of sugar. 5, 10, 15, 20, 25. That's what's in their control. That's what they've decided to manipulate or change. Right? And then the amount of alcohol depends on this, the amount of glucose. So the independent variable is the glucose, and the amount of alcohol, therefore, has to be the dependent variable. Now, I'm going to say this, and this is something that's very important. You have to remember the control treatment, 99% of the time, is going to be setting the independent variable to 0. So if you can figure out the independent variable and the dependent variable, then you can figure out the control. Usually. 0% glucose solution in this case, because the independent variable is glucose, a 0% glucose solution, in other words, putting yeast in a solution that has no sugar and letting them see if they can produce any alcohol. They shouldn't, because yeast shouldn't be able to grow just in water. They would need water and sugar. Okay, so that's the idea here. We're having a control treatment that allows us to compare. What three variables should be standardized? Remember, standardized variables are the ones that, in fact, have to be kept the same in all of the treatment levels. The treatment levels are the 5%, 10%, 15, 20, 25, and 30% glucose solutions. So if that's what I'm changing, these are the things that must be kept the same. The type of yeast, the amount of yeast, and the temperature of the solutions. Now, there are several other things that would also have to be kept the same. But I only wanted you to come up with 3. And those were the 3 that I thought of. If you thought of other ones and you want to check them with me in class, but be my guest. All right? I'm going to skip number 14, but I'm going to skip number 14. I'm going to move on to number 16 because that's a little bit harder. Okay, but number 14 is exactly the same as number 13, except for I didn't put an A, B, C, D, but you do the same stuff. All right, so I'm going to just skip ahead here to number 16 to see a little bit slightly different version, because this one's a little bit trickier. Do this one, I'm going to do number 17, number 18. We'll just talk about those. All right, so a team of scientists is testing a new drug, XYZ, on AIDS patients. They expect patients to develop fewer AIDS-related illnesses when given the drug, but they don't expect XYZ to cure AIDS. Okay, so it's a drug. It's hopefully going to help people with AIDS, but it's not going to fix it. What hypothesis are the scientists testing? Okay, so if I was you, I wouldn't necessarily start with the hypothesis. I would start with the independent dependent variable, because as we know, the hypothesis answers the question, how will the independent variable affect the dependent variable? Now, if you don't know what the independent and dependent variables are, then you can't really put a statement about those two things. So I would actually just start with, I'm going to get my purple here for my independent variable. I would start with that. So the question is, what are the scientists manipulating? What are they changing? Well, let's read the question. They expect patients to develop fewer AIDS-related illnesses when given the drug. Okay, so to me, the drug is the thing that they're testing. And then they're going to measure something about that drug on their patients. So the drug itself, XYZ, is the thing that they're manipulating. Now, it's not really clear in the question how they're going to manipulate that, but whenever you have a group of humans, you're going to have you're testing a drug on humans, you're going to have 2 groups. You're going to have a group that you give the drug to, and you're going to have a group that you don't give the drug to. Now, it wouldn't make a whole lot of sense to give a drug to one group and not to the other. So you kind of have to give a drug to both, but here's the key. Only one group is going to get the real drug. The other group is going to get a sugar pill, a placebo. It's not going to have any actual medication in it. And what you would look at as a scientist is you would measure what the differences between the real drug and the placebo were. Okay, so just going back to the independent variable, XYZ or no XYZ, that is the independent variable. All right, the dependent variable, if you look here, they expect patients to develop fewer AIDS-related illnesses. Right? That is your dependent variable. That is what the scientists are going to measure, because that's what the dependent variable is. Okay? Sorry. Go back here a little bit. Sorry, I checkmarked my hypothesis. My hypothesis then, if drug XYZ is given to patients with AIDS, they will develop fewer AIDS-related illnesses. So notice I have my independent variable, drug XYZ, and what I think the effect of that will be on the dependent variable. I've got that those both in there. Humans must, you can read over what the control treatment, we talked about that a little bit, and then there's 3 variables. With humans, it's almost impossible to get an exact. It's not like with plants. You can't just say, oh, here's 3 bean seeds and here's 3 bean seeds. I'm gonna use 3 in each. You can't find 2 people that are identical unless you use identical twins. It's very difficult to find that number of identical twins to test, especially for a study like this. So you're going to try and match up in your 2 groups, like we were talking about, the ones that get the drug and the ones that don't. You're going to try and match up as many things as you possibly can. So age, weight, relative health of patients to start, those types of things. And there'll be lots of others as well. All right, so now I'm going to move on to questions 17 and 18, which are also a little bit tougher. I'm going to show you how we answer those, and hopefully this will help you fill in your answers. All right, so question 17, again, it's a little bit tricky, but we're going to read it. A group of students decides to investigate the loss of chlorophyll in autumn leaves. They collect green leaves and leaves that have turned color from sugar, maple, sweet gum, beech, and aspen trees. Each leaf is subjected to an analysis to determine how much chlorophyll is present. All right. So first of all, you have to determine the independent variable. I'm going to tell you right now, there are two independent variables here. All right? There is the type of tree. As you can see, the scientists, oops, on here, that on. The scientists are changing, the types of trees, as you can see right there. So that's an independent variable. However, they're also collecting samples of different types of leaves. Leaves that are green versus leaves that are not green. Leaves that have turned color in the fall. So another one you can highlight is the type of leaf or the color of the leaf. Okay, so there's two. Most experiments don't have two. Okay, but this one does, so you kind of had to pick one. Question 17C, what is the dependent variable? Well, it doesn't matter which way you look at it. It's the amount of chlorophyll in the leaves. So they're going to measure the amount of chlorophyll in the leaves, the difference between the two, the green leaves versus the brown leaves, or the different trees, depending on which experiment you want to look at it from. So a hypothesis would be the leaves that have turned color will contain less chlorophyll than the leaves that haven't. Or if the leaves are from trees that have longer days in the autumn, then they will have a higher amount of chlorophyll. Yeah, maybe. I think that was one that another class gave me, so let's go back here, get my right color for my dependent variable here. Okay. Yeah, so I think personally I like the first one better. I think another class gave me that one and I accepted it, but I think the first one's a better one. Now, is the experiment a fair experiment? How might you change it to make it more scientific? Well, we talked about it in class. Generally speaking, you only have one independent variable and you measure one dependent variable. There are exceptions to that, and what you have to decide is if you think this experiment would work the way it's listed, or if it might be better to separate the two experiments and do them separately. Not really a wrong answer here, I just want you to think about it. Let's quickly look at question 18. So I'm going to move this up now. I'm going to erase that stuff. Where's my eraser tool? Erase that stuff. This doesn't work. I'll just undo all that. There we go. Okay, so I'm just going to move this up then and make it a little bit bigger. There we go. Okay, so what we're going to look at here, it's a slightly different version. A scientist wants to study mating behavior in crickets. She hypothesizes that males that win the most male versus male contests mate with the most females. She observes the crickets to obtain data. For each male, she counts the number of male-male fights he wins and the number of females he mates with. She experiments with 10 male crickets. Okay, so she hypothesizes that males that win the most male-to-male fights mate with the most females. So the independent variable then, the one that, again, this one she's not really manipulating, she's just setting it up, and the males are fighting each other, but it would be the number of male-to-male contests. That would be the independent variable, okay, because that leads to the ability to count how many females they get to mate with. All right? Now, would you recommend replication for this experiment? Why or why not? Well, I'm going to get you to go back and look at your notes on replication, which are back up in the booklet, and you can take a look at that. But I just wanted to point out a few of these things. Just one last thing before I let you go. Really, when you're doing independent and dependent variables, it's a very good idea to look at these sentences and underline things and highlight words that lead you to the right answers. And if you need some more help with that, please don't hesitate to ask me in class. Good night.